This is an A-level presentation on the structure of the cell surface membrane. Specifically, we're going to look at the arrangement of the actual membrane itself, the roles of the different parts within the membrane, and the structure of the membrane and its general role in and around the cell. Just to get started, an easy task. Just identify the structures that are found inside animal cells, plant cells, and bacteria cells. Please remember you're aiming for A-level versions here, so make sure you go over the relevant topics to include the A-level ones, not just the GCSE terms. Pause the video now, have a go, and then play the video when you're ready to go again. So in terms of animals, we have the following. Plants have the following, and bacteria, of course, have no membrane-bound organelles. It's one of the parts of a prokaryotic cell. Most of what we find inside a plant cell also found in an animal cell, but there are a few specific differences that you need to be aware of. Make sure you familiarize yourself with those. There are 5 main purposes to membranes. Please note the word membranes does not limit yourself to the cell surface membrane. This includes the membranes around organelles and in other places around the body. And the 5 main functions are, if you can use the picture to try and work it out, pause the video and play whenever you've had a go at it. So the 5 functions are: it controls what enters and leaves the cell, an internal transport system, isolation of enzymes, it provides surface reactions, and of course it separates organelles from the cytoplasm. The main cell surface membrane is made up of small subunits which are known as phospholipids. You do need to know the structure of a phospholipid. It has what's called the head end and tail end. The head end is the hydrophilic, which refers to hydro means water and loving, so it is attracted towards the water, whereas the hydrophobic end is repelled by water, and the cell surface membrane is orientated, so the hydrophilic parts are on the outside and the hydrophobic parts are on the inside. When we have the orientation, you'll notice that there's actually 2 layers of phospholipid. They actually have 2 layers sitting tail to tail. Now, this particular membrane, as is, does allow some substances through, but as it is a phospholipid, it will only allow lipid-soluble substances to diffuse in and out. Again, make sure you go back over diffusion. When you have water-soluble substances were not able to diffuse in and out. The membrane is also, if you imagine a giant ball pit, where the kids kind of get those adventure parks, and we've got all little plastic balls all laid out. Everything is flexible and movable, so you can put your hand in, move your hand around, and move your hand freely through the layer, but it keeps a constant seal as the balls reposition themselves in order to fill in the gaps. Now, the phospholipid bilayer, phospholipid of course being the molecule it's made from, and bilayer is because it is 2 layers. The phospholipid bilayer that makes up the cell surface membrane has a number of other things within it. And the majority of those other things that we're going to look at are types of proteins. And the proteins can fall into 2 main categories. You've got your extrinsic proteins, which are essentially on the surface of the cell surface membrane and held by weak bonds. And what you have then is the intrinsic proteins, which are held in place as they actually go into the phospholipid bilayer. If you notice on the diagram that the intrinsic proteins don't have to go all the way through, they just have to go into the layer, whereas the extrinsic are literally on the surface, and we've got very weak molecular forces, often ionic or hydrogen bonds, holding them in place, and so they're easily removable. So there are 3 main types of proteins inside the phospholipid bilayer. We've got our channel proteins, our carrier proteins, and our structural proteins. Now, if you look at the channel and carrier proteins, which is X and Z on the diagram, don't make the assumption that any particular shape automatically means that you have a channel or a carrier protein. Look at the information in the question and see what it tells you. For example, if it starts talking about ATP and it starts talking about active transport, then whatever protein it's referring to is going to be the carrier one. So don't just make the assumption that certain shapes mean it's an automatic channel or carrier protein. Look for information relating to the protein just to confirm your thoughts. So inside the phospholipid bilayer, we also have cholesterol. And cholesterol is there purely for a structural purpose. It's going to help reduce the fluidity of the actual cell surface membrane, and it also provides strength by pulling the fatty acids together. So the tails specifically get pulled together into little lumps. It also does have a role in preventing water loss and ions from the cell. So the glycolipids are connected to the cell surface membrane, or the phospholipid bilayer. Make sure when you see a diagram or a image taken from a cell and it asks what the thing sticking out of the cell is, you need to go back to its source. When you follow it back, if it's attached directly to the phospholipid bilayer, then it's a glycolipid. And glycolipids serve a number of functions. If, however, the thing sticking out is attached to a protein, an extrinsic protein, then what you have there is a glycoprotein, which has very similar structure or very similar functions to the glycolipids, but obviously is in a different location. So make sure that you know which one's the lipid and which one is a protein and how to tell the difference. All of that arrangement is referred to as the fluid mosaic model. The phospholipids can move among each other on a same plane, hence we refer to as fluid. And the mosaic is just because it's a random pattern. They're not evenly spread out, they're just randomly distributed throughout the entire cell surface membrane. Now there are two ways that substances can move in and out past the cell surface membrane. The first way is directly through the phospholipid bilayer. The second way is through either channel proteins or through carrier proteins. And it depends on the property of the particular molecule we're dealing with as to how it moves in and out of the cell. And just to finish off, a quick couple of questions, true or false, based on what you've seen during the lesson. At this point, pause the video and give yourself an opportunity to answer each of the questions. So the answers are false. The head is not referred to as a hydrophobic molecule. The head is hydrophilic. It will go towards water. The second statement is true. The third statement, the glycolipids attached to extrinsic proteins. Remember, if it's attached to a protein, it is a glycoprotein. If it's attached to the phospholipid, it is a glycolipid. Next one is true. Extrinsic proteins are also known as peripheral proteins. Active transport requires channel proteins. No, active transport is linked to carrier proteins and ATP. Small molecules will always move through a channel or protein channel. No, again, that's not always true because some can diffuse straight through the phospholipid bilayer. Water-soluble molecules can freely diffuse through the bilayer is false, and it has to be lipid soluble in order to get through the phospholipid bilayer, and channel proteins are filled with water is true. And that ends the presentation.